Hello, everyone. Thanks for joining us. Please, if someone can tell me if you can listen to me okay, and if you can see the screen. So, because I was having some troubles with the connectivity. Perfect. So, thank you for joining us. Welcome to a new Conda Lab talk. Today we are going to talk, as the name says, about the PCR. And like it's one technique with the southern applications. We are really happy with our conduction line, that is the qPCR line from Conda Lab. And today we will also introduce some new kits for allergen detection. So thank you for joining us and taking the time to be in this webinar. I will introduce myself. My name is Pilar Rodriguez. I'm the Rapid Microbiology Product Manager at Conda Lab, and I'm especially focused in our conduction line that we have been working for a few years, but today we will introduce some new kits, and well, we are really happy because the PCR is a technique that is becoming more famous and more with more applications every day, so we are really happy to bring you more solutions. So it will take us around 20-25 minutes only, and if you have any question or any doubt, you can write it here in the go-to application, and at the end I will try to answer everything. And of course, if you want, you can write us an email, and we will try to answer all of your questions. So I will turn off my camera so we don't have any connectivity problem, and we will start. So, sorry. Today we are going to just a brief review about the principle of these techniques, then see how to implement the technique and which furnitures, which equipment and everything we need to have in the lab. Then moving to allergen detections, and at the end we will talk just about all the conduction solutions. So to start, with the basic principle of the PCR. The PCR is a molecular biology technique that became really famous in the pandemic but was developed a few years ago. The main objective of it is to amplify a specific fragment of DNA. So if, for example, we are looking for Legionella, we will amplify a specific fragment, a really little specific fragment that is only present in Legionella. So this basically involves a pair of primers that will stick to the DNA strain, and by the use of a polymerase, we can amplify it. All of this happens in the thermal cycler, that is the PCR machine, and it happens just in 3 steps. First, the denaturation of the DNA strains, then the annealing, that is when the primer sticks to the DNA, and then the amplification, that is the when the polymerase act. All of this happens in around 30 or 35 cycles, and it's just different temperatures. Our kits are a real-time PCR, so we have two different PCR, conventional PCR and real-time PCR. Even though they have the same steps, the main difference between them is that real-time allows us to amplify and quantify simultaneously by the use of fluorescent dyes that will bind to the target DNA. The results, we will see it as in this picture. If we have a logarithm curve, we can say that is a positive result because there is fluorescence, so it means that we amplify something. And if we don't have a curve, it means that is a negative result. The equipment has a fluorescent sensor, so the equipment automatically gives us these kind of graphics. Talking now about how to implement the technique in the lab, sometimes there is like some afraid of this technique because people think that you will need a new lab on or a big space, or sometimes that you will require a lot of equipment. And the most important thing in PCR is to think about the workflow. To think, because we cannot, we have to be really careful with contamination. So it's important to think about the workflow and how the sample has to move in the lab. First of all is the reagent preparation. That is just mixed two reagents is really easy, but it has to be really careful because if we contaminate these reagents, we will have to throw it away. So it has to be really careful. For this, we don't need a lot of space because, as I already said, you just have to mix two reagents. The volumes are really small, so with a little space, it's okay. The cabin is an optional equipment only if in your lab you have. It depends on how you structure your lab. In some case, you will need a cabin to just to protect the regions. The DNA extraction step is like the dirty step because you will work with the real sample and you will have the microorganism that you are looking for, but you will also have everything that that sample contains. So you have to have a specific space for that. It doesn't have to be a different lab, just a table that you will only use for DNA extraction. For this, you will need a centrifuge, a thermoblock, and a vortex. And the cabin is optional, and it depends if you are working with a pathogen that can be dangerous for the technician that is working. If it's not, you don't need a cabin. And then the detection part, the qPCR step. The most important thing is the thermal cycler, that is the PCR machine that I already told you. Um, and here also we have to be careful to no contaminations. And if you have a big laboratory with a lot of samples, we also recommend to have a plate centrifuge or a mini centrifuge to, but it's not really necessary. It's just an accessory. The most important thing here is to think of the workflow to avoid contaminations and to have reliable results. And talking about the thermal cycler, in Condalab we have the CDL96, that is a thermal cycler. It's an open system. That means that you can use it with our kits, but you can use it also with other kits from the market. Or if you have your own designs, you can also use them with this equipment. It has capability for 96 samples. If you want, you can run the 96 samples, or you can run 10, 20, whatever you want. It has a high sensitivity and precision. The software is really intuitive, it's really easy to set up the programs. It's an equipment that is really friendly for the user. Also contains a data analysis system, and it has 4 channels of fluorescence that are the most common one. That is FAM, X, ROX, and CY5. But also, if you work with the other 2 channels, you have the possibility. We have the possibility to have it for 6 channels. The most important thing of this equipment that is not too big. Is really I use it a lot, so I know that it's really easy and really friendly, really intuitive to set up. So I really like it. Talking about when to apply this technique and thinking of a process, is a technique that is really flexible and you can adapt it to all of the matrix that you work for. If we think about an industrial process, we can say that we can apply it in the raw material control. We can apply it for contamination control in equipment, and also in intermediate product control and in final product control. These techniques allow us to Also, if we have a contamination in the process, with this technique we can test all the steps of the process and find where is the contamination. Also, with this technique we have, with the same platform, we can detect different types of microorganisms. I mean, we can test pathogens, for example, salmonella or listeria. We can also test virus, fungi, parasites. We can also test allergens or GMOs. So it's with the same equipment and the same lab, we can test all these kind of microorganisms. So we can, for example, if we produce, I don't know, food, but in the process we use water, and we also have to do the Legionella test for the water. We can use the PCR for that, and also for the control of the final product. So we can adapt it to all of the steps. Also, we can work with, it's a technique that allows us to work with different matrices, because if you choose the correct DNA extraction kit, you can test every kind of matrices. So it's a technique that is really flexible, and we can apply it in every steps and every kind of matrices. Moving to allergen detection now. There are two technologies that are really common for allergen detections, that are the rapid test and the ELISAs. These techniques are really common. Both of them are immunotechniques. The rapid test, well, you may know it because it's being used for everything. It's really simple and really easy. If you have two lines, you can say that is a positive result, and if you have one line, it's a negative result. But the problem with these kits is that the specificity and the sensibility is not too high. And then there is the ELISA kits, that these work with antibodies, that the sensitivity and specificity is higher than a rapid test. But also there are some gaps that this kind of kit cannot fill. So here is when the PCR comes to try to fill those gaps that this technique can't. If we talk about the advantages of this technology, we can start with specificity. As I said in the beginning, the PCR amplifies a specific fragment of DNA. So we are looking something that is only in that kind of allergen, if we are talking about allergens. And with the ELISAs or with the rapid test, sometimes the allergens are really similar. So the antigens or the antibodies are the same. And this kind of kit cannot differentiate between which one is it. And with with the PCR, we can do it. Then it's a technique that is really used for confirmation, because, well, the rapid results, and because the specificity and also the sensitivity of the test, of the technique, sorry. Also, we have a large catalog. There are some kind of allergens that don't have an ELISA or an lateral flow now in the market, so we can fill that with the PCR. And also this technique allow us to do a simultaneous detection. For example, if in one sample we want to detect three different kind of allergens, we can do it at the same time. That is something that we cannot do with the other kind of technologies. This is our I'm sorry. This is our catalog for allergens. We have 11 kits. All the kits came in the same way for 100 reactions. And all the kits, what is really good, that they have the same programming for the PCR, so you can run them all at the same time. If, for example, for one sample you want to test, I don't know, almond, hazelnut, and peanut, you can do it, the three of them at the same run in the thermal cycler. we are really really happy with these new kits because I think that is a really interesting and a really good technology to fill these gaps that the other technologies have. So well, that that we are really happy with it. Moving to our contagion solutions, these are all our kits we have for pathogenic bacteria as Salmonella, Listeria, Campylobacter, STEC. We also have we have three kits for water control for Legionella spp, Legionella pneumophila, and a multiplex that detects both of that. And then we have two kits for spoilage for beverage industry. And well, these kits, as the allergen kits, they are all for they all came, sorry, in the same way with the positive and negative control, and also the manipulation is minimal because it only are two regions that you have to mix. Our line has some competitive advantages. All our kit follow the PCR requirements that says the ISO. All of them have an internal control of the process. That means that in each well that you will run, you will have an internal control that tells you that the enzyme works okay, that the thermal cycler works okay, so that you are sure that the results are correct. And also a positive and a negative control. We also have some additional security measures. We have all the kits have an UNG control for cross-contamination. And we also have a viable cell vision. This vision is to avoid false positive results that come from dead cells. That is a PCR problem. So we have this kit available too. Then all of our kit can be transported at room temperature. When you receive it at your lab, you have to store it at the correct temperature, but we can do the shipment at room temperature. And well, as I already said, a minimal manipulation because the protocols are really simple. All of our kits have homogeneous cycles. As I already said, you can run different kind of pathogen or microorganism at the same time. That this makes you save a lot of time in the lab, and it's an open system. The thermal cycler, you can use it with other brands, and also our kits can be used in another thermal cyclers that are also open. So as a conclusion, what I think the most important thing that I want to say is that these techniques allow us to have a lot of opportunities and optimize different kind of process because we can detect a wide variety of matrices, a wide variety of microorganisms. So we can use it in each step of the process of our process. Depends on the industry. It has a lot of benefits as speed, specificity, and more. So maybe the investment is higher compared to traditional techniques, but we can use it in each step and we can have a lot of benefits. So well, that's all. Thank you all for joining us. Here is a QR code that you can scan and will to answer some question about this webinar. And if you have any comments or if you, I don't know, want another webinar about something else, you can leave it here, your message. And if you have any question, you can write it here and I will try to answer. For every kit, we have the validation report and the instruction for use that we can send you. Here Shuri is asking about the sensitivity. We can send you the technical data sheet with all those parameters. So if you want, we can you can send us an email and I will send you the validation report with all that information, Shuri. Here also asking about the extraction kits. We also have some extraction kits that are for DNA extraction kits that are for food industry. That depends on the matrix that you are working on. You have to choose one or other. Okay, well, if you don't have any other question, as I said at the beginning, you can send us an email with any doubts or any other question, and I will be happy to answer all. And thank you for joining us in this webinar, and I hope to see you again. So thank you everyone for joining.